Have you ever just stopped and thought about how some living things, some organisms seem to change and adapt, overcome these huge challenges at a pace that's almost, well, bewildering? When we think evolution, usually our minds go to eons, right?
Yeah.
Slow, gradual processes over thousands, millions of years.
Right, long game.
Exactly. But then you look at bacteria, these tiny single-celled things, and they seem to have these amazing shortcuts.
They really do.
1 minute, a strain is vulnerable, easy to kill with an antibiotic. The next, it's like a super resistant tower house. It feels like it happens overnight.
Yeah, that observation, it really hits the nail on the head. It's a powerful testament to just how incredibly efficient bacterial genetic variation can be. Almost unparalleled, really.
Yeah.
Unlike more complex organisms, which, mostly rely on those slow random mutations being passed down generation by generation. Bacteria have these sophisticated direct methods, ways to grab entirely new genetic traits. So this deep dive, it's all about unpacking how they manage this rapid genetic change. We're really going to focus on gene transfer, how they share that genetic code.
Okay, let's definitely unpack this. It's fascinating. Imagine like a world where crucial information, the actual instructions for survival, spreads like wildfire. Not like emails or tweets, but actual physical genetic code. And this swap turns, humble bacteria into these super survivors. It's like watching evolution on fast forward.
Exactly.
So our mission today, really get to grips with these ingenious and, sometimes pretty alarming ways bacteria show their secrets.
Precisely. We are embarking on a comprehensive deep dive here into this captivating world of bacterial genetic variation. We'll be focusing hard on the 4 main mechanisms of what we call horizontal gene transfer. Conjugation, transduction, transformation, and transposition.
Okay.
And we'll also look at the really crucial roles of plasmids, these mobile DNA elements, and recombination, which is how the new DNA gets integrated.
Right.
Our goal, really, is to give you a crystal clear, thorough understanding of these processes. And crucially, why they matter so much, especially when we think about medicine, pathogen evolution, and, the huge challenge of antibiotic resistance.
Okay, so to really set the stage properly here, let's first lay out the 2 main ways bacteria get genetic variation.
Good idea.
On one side, you've got mutation. That's the classic idea, right? A random, often quite slow change in the bacterium's own DNA.
Yeah, like a typo in the instruction manual.
Exactly. A little tweak here or there. Over lots and lots of generations, maybe these tiny changes add up to something useful. But then, there's the other way. Horizontal gene transfer.
The game changer.
Totally. This isn't just tweaking the existing DNA. This is acquiring whole new chunks of pre-made functional DNA from somewhere else.
Right.
It's not editing the manual. It's like instantly downloading a whole new software package, maybe even a whole new operating system, right into their existing setup.
Precisely. And what's truly fascinating, what makes this so powerful, is that difference. Think about it. It's like the difference between spending years slowly practicing a skill versus, instantly downloading fluency in a language.
Right. Or knowing how to do complex surgery just like that.
Exactly.
Yeah.
With horizontal gene transfer, a bacterium can suddenly gain a really complex ability it never had. Maybe resistance to multiple drugs or the power to make a potent toxin.
Wow.
And it doesn't have to wait for that incredibly unlikely series of random mutations to happen in its own family line. It's a true evolutionary shortcut.
So they're borrowing superpowers.
Essentially, yes. Borrowing, inheriting traits from neighbors, sometimes even across different species. This lets them react incredibly fast to pressures like, say, a new antibiotic in their environment, with a speed that, honestly, really challenges our ability to control them. It fundamentally changes the whole landscape of how microbes evolve.
Okay, so when we talk about these amazing shortcuts, the first one we absolutely have to dive into is conjugation. I kind of picture this as the bacterial love story, although, let's be honest, it's probably more about survival than romance.
Chuckles slightly.
Yeah.
That's a fair way to put it. It's about direct contact, isn't it?
Yeah, direct physical contact seems like a really efficient, precise way for them to share their most important genetic stuff, like 2 cells literally linking up cell to swap vital info.
That analogy really captures the essence. At its heart, conjugation is this direct, one-way transfer of genetic material, usually DNA, from a donor bacterium to a recipient, and happens through this temporary physical bridge.
Okay.
The whole elaborate process is typically run by a special piece of extra DNA called a fertility plasmid, or F plasmid.
Ah, the F factor.
Exactly. And this F plasmid isn't just cargo, it's the conductor. It carries all the genes needed to make the transfer happen, including, crucially, the instructions for building that physical connection. That's the sex bolus.
The sex bolus, right. So let's try and picture this happening, down at the microscopic level.
Okay.
You've got the donor cell, the F plus cell, because it has that F plasmid.
Correct.
And it actively sticks out this sex pilus, which isn't just a random bit sticking out, right? It's more like a grappling hook.
Very much so. It's dynamic. It actively seeks out and latches onto specific spots, receptors, on the surface of a recipient cell, the F cell, which doesn't have the plasmid.
And then what? Does it just hold it there?
No, it actually retracts. It reels in the recipient cell, pulling them into direct, close contact. That creates a stable channel, the conjugation bridge, for the DNA to pass through. It's a very active, directed action.
Wow. Okay, so they're physically connected, then the DNA transfer happens.
Precisely. It's more than just a passive link. Once that bridge is solid, the circular F factor DNA inside the donor gets nicked by an enzyme at a specific spot, the origin of transfer, or ORIT.
Okay.
Then one strand of that mixed F factor DNA unwinds and is carefully fed, single file, across the bridge into the recipient cell.
Just one strand.
Just one strand initially. But here's the clever bit. To make sure both cells end up with a complete double-stranded plasmid, a new complementary strand gets made simultaneously in both cells, using the transferred single strand and the remaining single strand as templates.
Ah, so they both end up with a copy.
Exactly. You start with one F plus and one F, and you end up with two F plus cells. Both now have the F plasmid, ready to go out and conjugate again.
That answers the question. I was just thinking, why would the donor give away its plasmid? It duplicates it.
It duplicates it, ensuring its own survival and spread. It's incredibly efficient. And that new F plasmid in the recipient, it can even integrate itself into the main bacterial chromosome, becoming a permanent fixture.
And you mentioned it's fast.
Relatively speaking, yes. Transferring even the entire bacterial chromosome, which is much larger, can take about 100 minutes under good conditions. In evolutionary time, that's lightning fast.
That is fast. And what's really cool is that conjugation isn't just about passing the F plasmid itself, right? There are variations.
Absolutely. It's more nuanced than just that simple transfer. The most common type, as we've discussed, is that straightforward plasmid transfer. F plus to F. Very efficient. Quickly turns recipients into donors.
Okay, basic model. What else?
Well, then you get chromosomal gene transfer. This happens if the F plasmid, instead of floating free, actually integrates itself into the main bacterial chromosome.
It becomes part of the main genome.
Exactly.
Right.
And that cell is now called an HFR strain, high-frequency recombination.
HFR. Okay.
Now, these HFR cells can still conjugate. They can start transferring DNA, but because the F factor is now part of the chromosome, they start transferring chromosomal genes along with it.
So they can share their main genes, too.
They can, but here's the catch. The whole chromosome is huge compared to the plasmid. That conjugation bridge is often quite fragile. It frequently breaks before the entire chromosome transfer is complete.
Ah, so the recipient might only get a piece?
Often just a piece, yes. Leading to what we call partial diploidy, having two copies of some genes, but not others. Still, it's a powerful way to move chromosomal DNA around.
Okay, that makes sense. And then you mentioned a third type, the really interesting one.
Ah, yes, F prime. F transfer, sometimes called sexduction. This is where it gets particularly clever, showing how flexible these systems are.
Sexduction, I like that.
Chuckles. Imagine that integrated F factor in an HFR cell decides to leave the chromosome. But when it cuts itself out, the excision isn't perfect.
It makes a mistake.
Kind of.
Yeah.
It accidentally snips out a small adjacent piece of the bacterial chromosome along with its own F factor DNA.
So it grabs some neighboring genes.
Precisely. Now you have this F prime plasmid. It carries the F factor genes plus some specific chromosomal genes.
Right.
And this chimeric plasmid can then be transferred super efficiently, just like a normal F plasmid, to a new recipient.
Wow. So the recipient gets the F factor and those extra chromosomal genes all in one go.
Exactly. It's like moving house and accidentally packing a few valuable things from your neighbor's shed along with your own stuff and then giving them to your new neighbor.
That's a great analogy. Very efficient, if a bit sneaky.
It's highly effective for moving specific blocks of genes. Now, it's also important to remember, conjugation isn't just for gram-negative bacteria with their fancy pili.
Oh, right. What about gram-positives?
Gram-positive bacteria do it too, but they have a different approach. No pili. Instead, the donor cells produce these sticky surface proteins called adhesions.
Adhesions?
Yes, these allow them to clump together directly and tightly with recipient cells, forming aggregates that let the DNA transfer happen.
So more of a huddle than a bridge.
A bit like that, yeah. And some cases are even more fascinating. Certain Streptococcus species, for instance, the recipient bacteria, actually produce chemical signals, sex pheromones.
The recipients send out signals, like an invitation.
Almost exactly like that. They signal they're ready to receive DNA, basically initiating the process from their end. It shows this amazing level of communication and coordination.
That's incredible. Okay, so we've got the mechanisms, the variations, but let's bring it back. Why should we care so much about conjugation? What's the big deal clinically?
Oh, the clinical significance is. It's immense.
Oh.
Profoundly critical, actually.
Why is that?
Conjugation is arguably the single most important mechanism for the rapid, widespread spread of antibiotic resistance among bacteria.
The main way resistance spreads.
Very often, yes. This resistance is frequently carried on those R plasmids we mentioned, plasmids loaded with genes that neutralize antibiotics.
Okay.
And because conjugation is so efficient cell to cell, a single resistant bacterium can quickly share its entire resistance toolkit with many neighbors, even across different species, especially in places like hospitals where bacteria are under intense antibiotic pressure.
So that's how we get these superbugs.
It's a primary driver behind the rise of multi-drug resistant superbugs. Absolutely. It's why treating infections is becoming so much harder. But it's not just resistance.
What else?
Conjugation also spreads genes for making bacteriocins, toxins that kill competing bacteria, often on cofactors. It spreads genes for heavy metal resistance, letting bacteria survive in polluted places. It transfers genes for making pili, which help bacteria stick to our tissues and cause infections.
Sticking power. Okay.
And, crucially, it spreads genes for making exotoxins, nasty secreted proteins that directly cause disease symptoms and damage our bodies. It can turn a harmless bug into a serious pathogen.
So putting it all together, the bacteria causing infections aren't just passive targets.
Not at all.
They're actively networking, sharing survival tips, weapons, defenses, like a microscopic intelligence agency, constantly upgrading their abilities right under our noses, even inside a patient.
That's a very apt description. It's a dynamic, incredibly effective sharing network. Conjugation is one of their most powerful tools in this ongoing arms race, a real game changer in their world.
Okay, wow. Let's shift gears then. Another mechanism. Transduction. You said this one is different, indirect, involves viruses.
That's right. This method uses a middleman, often an unwitting one. Bacterial viruses, which we call bacteriophages, or phages for short.
Phages. Okay, so how does a virus end up delivering bacterial genes?
It's a fascinating, somewhat accidental process. It happens during the phage's replication cycle inside a host bacterium. The virus hijacks the cell's machinery to make copies of itself, its own DNA, its protein.
Right, the usual virus routine.
But sometimes during the assembly of new phage particles, a piece of the bacterial host's DNA gets mistakenly packaged into a new phage head, instead of, or sometimes along with, the phage's own DNA.
The virus makes a packaging error.
Exactly. Then when this transducing phage, carrying bacterial DNA, infects a new bacterium, it injects that bacterial DNA it picked up, not its own viral genome.
Ah, so it delivers the wrong package.
Precisely. And that transferred bacterial DNA can then get integrated into the new host genome through recombination, giving it new traits.
Okay, here's where it gets really interesting for me. That analogy you use, the delivery drone picking up the wrong package, like grabbing a briefcase of secret documents instead of medical supplies and delivering that somewhere else, that's basically what the phage does.
That's essentially it. An accidental genetic mix-up, but one with potentially huge consequences for the recipient bacterium, suddenly giving it abilities it never evolved itself.
Amazing. And like conjugation, are there different types of transduction?
Yes, there are two main types, based on what DNA gets packaged and how. First, there's generalized transduction.
Generalized, meaning random.
Pretty much. It occurs when the phage can potentially carry a segment from any part of the bacterial chromosome. This usually happens with olytic phages, the kind that replicate and boost the host cell open.
Okay.
During that process, the host DNA gets chopped up into fragments. If a bacterial DNA fragment is roughly the same size as the phage genome, it can get accidentally stuffed into a phage head during assembly. So any bacterial gene has a roughly equal, albeit small, chance of being transferred.
Random pickup. Got it. What's the other type?
The other is specialized transduction. This is more specific, more targeted.
How so?
This involves temperate phages viruses that can integrate their DNA into the host chromosome and just sit there quietly for a while, becoming a prophage.
Like a dormant spy.
Exactly. It replicates along with the bacterial DNA. But later, if the prophage gets activated maybe by stress and cuts itself out of the chromosome to start making new viruses, sometimes that cutting process, that excision, is sloppy.
Another mistake.
An imperfect excision. It accidentally takes an adjacent piece of the bacterial chromosome with it.
Ah, so only genes right next door to where the phage was hiding.
Precisely. That's why it's specialized. Only bacterial genes located very close to the phage's integration site get transferred. It's less random, but can be very efficient for moving specific genes linked to the phage's location.
Okay, that makes sense. Now, you mentioned something called lysogenic conversion. That sounded important.
Extremely important. It's a major consequence, especially of specialized transduction, and a real game changer for the bacteria.
What is it exactly?
It's when the integrated prophage itself, the phage DNA sitting in the bacterial chromosome, actually gives the bacterium new properties, often traits that make it more harmful, more virulent.
The virus DNA itself makes the bacteria dangerous.
Sometimes, yes. The phage isn't just a delivery truck. Sometimes the phage genome itself carries the genes for virulence factors.
Wow. Can you give some examples?
Absolutely, and they're clinically very significant. Think about diphtheria, the potent toxin that causes diphtheria. It's only made by Corynebacterium diphtheria if it's been infected by a specific phage. The toxin gene is on the phage.
No phage, no toxin.
Essentially, yes. Same for the botulinum toxin,
Mm-hmm.
an incredibly dangerous neurotoxin causing botulism. It's encoded by a phage gene in Clostridium botulinum, and the erythrogenic toxin in Streptococcus pyogenes that causes the scarlet fever rash. Phage gene again. Perhaps the most famous example today is the Shiga toxin in E. coli 0157-H7, the dangerous foodborne pathogen. That toxin production is also due to lysogenic conversion by a phage.
So these aren't just minor tweaks. These bacteria become seriously dangerous or pathogenic at all because a virus moved in.
Exactly. It highlights this incredible interplay. The virus brings the weapon. What's really fascinating now, almost paradoxical, is that we're exploring using these very phages and phage therapy to fight antibiotic-resistant bacteria.
Using the virus to kill the bacteria, it sometimes empowers.
Precisely. It's a complex relationship.
It really is. So, thinking clinically again, transduction seems widespread and pretty impactful too.
Undeniably. It plays a huge role in spreading antibiotic resistance, just like conjugation.
Yeah.
For example, penicillinase genes, which break down penicillin, are often passed between Staphylococcus aureus strains by transduction. That contributes to things like MRSA. If you look at the big picture, transduction is recognized as one of the main ways genetic material moves between bacteria and nature. It's happening constantly, everywhere, in soil, in water, in our own gut.
It's a constant shuffling of genes.
A constant shuffling, yes, driving adaptation, driving resistance, and presenting us with ongoing challenges in controlling infections. It's a really powerful, pervasive force.
Okay, let's pivot again. Mechanism number 3, transformation. You describe this one as a direct uptake of naked DNA, like DNA just floating around.
That's the core idea. Transformation naturally happens when bacteria die and lyase, spilling their guts, including their DNA, into the environment.
So DNA debris.
Essentially, yes. Now, other living bacteria nearby, if they're in a special physiological state called competence, can actually grab this free-floating naked DNA from their surroundings.
Competence. They have to be ready for it.
Exactly. It's a regulated state.
Right.
Often triggered by things like stress, nutrient shortage, or high population density. The competent cells produce special proteins on their surface that bind to the DNA and transport systems to pull it inside.
Okay.
Once that foreign DNA is inside, it can then potentially recombine with the recipient's own chromosome, integrating new genes.
So I like your analogy of the scroll washed ashore.
Mm-hmm.
It's out there, but you have to be ready and able to grab it and understand it for it to be useful.
Yeah.
If you're not competent, it just drifts away.
That's a perfect way to visualize it. And the discovery of this process was actually a massive moment in biology.
Oh, really? How so?
It was first observed back in 1928 by Frederick Griffith, working with Streptococcus pneumonia, the pneumonia bacterium.
Griffith's experiment, right?
Exactly. He showed that harmless, non-virulent bacteria could become virulent, deadly, just by being mixed with dead virulent bacteria. Something was being transferred.
The transforming principle.
Precisely. It was a huge puzzle. What was this substance that could fundamentally change a bacterium's nature?
And the answer turned out to be DNA, right?
Huh.
That was Avery, McLeod, and McCarty.
Yes, in 1944. Their incredibly careful work proved conclusively that the transforming principle was DNA, not protein, which many scientists had suspected.
A landmark discovery.
Absolutely foundational. It established DNA as the genetic material, paving the way for basically all of modern molecular biology and genetics, all from observing bacteria picking up DNA from their dead neighbors.
Incredible. So how common is this in nature? Does it cause a lot of disease spread?
Well, while it was historically crucial for discovery, natural transformation seems to happen reliably in only a relatively small number of bacterial types. Genera like Bacillus, Haemophilus, Neisseria, Acinetobacter, and some Streptococcus and Staphylococcus species are known to be naturally competent.
So not universal.
Not at all. And its actual contribution to spreading disease or resistance genes in the wild is generally thought to be pretty insignificant compared to conjugation or transduction. It happens, but it's likely not a major driver of epidemics, for instance.
You mentioned it's huge in the lab.
Yes. That's the crucial distinction. Despite its limited role in natural disease spread, transformation is the workhorse technique in genetic engineering.
How do scientists use it?
We use it constantly to introduce specific pieces of DNA into bacteria, especially E. coli. We can artificially make bacteria competent using chemical treatments or electrical pulses electroporation, forcing them to take up DNA we provide.
So you can give them specific genes you want them to have, like for making insulin.
Exactly. Insulin, other therapeutic proteins, fluorescent markers to track them, countless applications. So, while maybe not the big bad wolf in nature, transformation is an absolute superhero in the lab, a fundamental tool that enabled the entire biotech revolution.
Okay, fascinating contrast between nature and the lab. Right, let's move to the fourth mechanism, transposition. You called these jumping genes. How does that work? Sounds different again.
It is quite different because it's primarily about movement within a single organism's genome. We're talking about specific bits of DNA that can move from one spot to another.
Jumping around inside the cell's own DNA?
Exactly. They can hop from one place on the chromosome to another, or jump from the chromosome onto a plasmid, or between plasmids within the same cell.
How do they jump? Do they need matching sequences, like recombination?
That's a key difference. They generally don't need much sequence similarity, much homology between themselves and where they land. They have characteristic sequences at their ends, and they often encode their own enzyme, transpose S, which recognizes these ends and basically cuts and pastes the transposon into a new location.
So they bring their own cutting and pasting tools.
Pretty much. And often it's a copy of the transposon that moves, leaving the original behind. This is called replicative transposition, so the transposon duplicates itself as it moves. Other times it's a simple cut and paste conservative transposition.
Okay, and the simplest ones are just the jumping machinery.
Right, those are called insertion sequences. They basically just contain the transposis gene and the necessary signals for movement. More complex transposons carry extra genes between those signals.
And you mentioned they're different from plasmids because they can't replicate on their own.
Correct. That's crucial. Unlike plasmids, transposons rely entirely on the host's replication machinery, the chromosome or plasmid they happen to be sitting on, to get copied.
So, like, that internal relocation team, they have the tools to move stuff, but they need the house's power supply, the host's replication system, to actually make copies.
Excellent analogy. They're movers, not self-sufficient replicators.
Okay, so what's the clinical significance of these internal jumpers?
Oh, they're incredibly important medically.
Yeah.
Transposons are very effective vehicles for spreading important traits within and between different DNA molecules in the cell. They frequently carry antibiotic resistance genes.
So they can move resistance from a plasmid to the chromosome or between plasmids.
Exactly. This makes resistance genes highly mobile within the bacterial genetic landscape. They also carry genes for metabolic enzymes, maybe allowing bacteria to eat new food sources, or genes for toxins, boosting virulence.
So they spread useful tools around internally.
Yes, but their jumping also has other effects. When a transposon lands right in the middle of a functional gene, it can disrupt it, basically causing a mutation and knocking out that gene's function.
An insertion mutation.
Right. Or if it lands near a gene, its presence can actually change how that nearby gene is expressed, maybe turning it on higher or lower or switching it off completely. This can have unexpected consequences, potentially activating a silent gene or silencing an essential one.
So these jumping genes are like stealthy internal agents of change.
That's a good way to think of it.
They're not bringing in outside info, but they're constantly rearranging the bacterium's existing toolkit from the inside out. moving skills around, sometimes breaking things, sometimes activating new abilities.
Precisely. This internal reshuffling can dramatically impact the bacterium's survival, its disease potential, and its response to treatments. It's a constant dynamic rewiring of the internal blueprint.
Okay, we've covered these 4 incredible mechanisms: conjugation, transduction, transformation, transposition. And one word kept popping up again and again: plasmid.
Can't talk about bacterial gene transfer without them.
Right. So let's bring our focus back squarely onto plasmids. They seem absolutely central, acting as carriers, connecting to conjugation, moving transposons around, even relevant for transformation in the lab.
They are indeed central players. Their versatility is remarkable. So formally, plasmids are extrachromosomal, meaning outside the main chromosome, typically double-stranded circular DNA molecules.
Extrachromosomal, circular DNA. Got it.
The key thing, their superpower, really, is that they can replicate independently of the main bacterial chromosome.
They copy themselves.
Yes, using the cell's machinery, but on their own schedule. They can exist in multiple copies within a single cell, sometimes just a few, sometimes hundreds.
Wow. Are they essential for the bacteria to live?
Generally, no. A bacterium can usually survive fine without a particular plasmid, if conditions are easy. But, and this is a crucial part, plasmids very often carry genes that give the bacterium a significant advantage under specific pressures.
So they're like optional upgrades, genetic accessories.
That's a perfect description. Optional, but incredibly useful accessories that can be picked up, swapped, and used for survival and competition.
And we've touched on the types already: the F factor for conjugation.
Right, the fertility plasmid.
The R factors for resistance, which sound like a huge clinical problem.
Absolutely huge, carrying resistance to often multiple antibiotics.
And Col factors for making those bacteriocins the bacterial weapon.
Exactly, killing off competitors. So plasmids really equip bacteria for the tough microbial world.
The clinical importance seems massive then.
It simply cannot be overstated. Plasmids are just incredibly important in medical microbiology. They frequently carry these genes for advantageous traits. Antibiotic resistance is the most notorious, obviously.
Yeah.
But also resistance to heavy metals, mercury, silver, things found in hospitals or industrial pollution. Genes for making pili, which we said help them stick to us, or medical devices, promoting infection and biofilms.
Right, sticking power again.
And very importantly, genes for making various exotoxins, secreted poisons that directly cause disease symptoms, from diarrhea to paralysis.
So it makes me wonder, how do these relatively small optional DNA circles wield so much power?
It's their combination of features, really. Their mobility, they can be easily transferred, the fact that they can carry multiple potent genes together on one unit, and their ability to replicate to high copy numbers, amplifying those genes. They are dynamic, powerful players in what we call the bacterial mobilome, the entire collection of mobile genetic elements. And they contribute massively to bacterial pathogenicity and, yes, to the enormous challenge of treating infections.
So summing it up, plasmids are like the ultimate bacterial accessory, letting them quickly grab, copy, and share traits that make them tougher, more competitive, often much more dangerous.
That's it. Yeah.
Their ability to spread these traits, resistance, virulence, is a huge factor in why bacterial infections remain such a major global health threat. Okay, so we've seen DNA get delivered, handed over by conjugation, dropped off by transduction, picked up by transformation. But what happens next? How does that new DNA actually become part of the bacterium's working set of genes?
Ah, yes, the final crucial step. That's where recombination comes in. It's the process that integrates the newly arrived foreign DNA into the recipient's genome, making it stable and functional. The assimilation step.
Recombination, okay.
So, regardless of how the DNA got in conjugation, transduction, transformation, once it's inside the recipient cell, it often needs to be physically incorporated into the host's DNA to be reliably passed on to daughter cells and properly expressed.
Otherwise, it might just get lost or broken down.
Exactly. Recombination prevents that. It's basically a molecular cut and paste process, exchanging DNA segments. There are 2 main ways it happens. The most common is homologous recombination.
Homologous meaning similar.
Yes, it requires extensive similarity in the DNA sequence between the incoming foreign DNA and a region on the host chromosome, like matching puzzle pieces. The cell has machinery involving proteins like Rha that recognizes these matching sequences and carefully swaps the pieces, integrating the new DNA.
Okay, like fitting a new compatible piece into the puzzle. What's the other way?
The other main type is non-homologous recombination. This one is different because it needs little or no sequence similarity.
So it can stick DNA in almost anywhere.
Pretty much.
Oh.
It's more like forcing a new, differently shaped piece into the puzzle. It uses different cellular machinery, often associated with mobile elements like transposons. It's less precise, sometimes more disruptive, but allows for integrating DNA that doesn't have a matching spot.
Okay, so connecting this back to the big picture, recombination is really the key step that makes the transferred DNA stick, makes it a permanent working part of the bacterium.
Absolutely critical. It's the transition point.
Right.
Without successful recombination, that transferred DNA is often just transient, unstable. It might get degraded or lost when the cell divides. It wouldn't provide a lasting benefit.
Right.
It's through recombination that the bacterium truly incorporates those new capabilities, antibiotic resistance, toxin production, a new metabolic pathway, making them a stable, inheritable part of its identity, ready to be expressed and used. It's the final act of genetic assimilation, turning borrowed code into owned functional traits. That's what really drives the adaptation and evolution we see.
Wow. Okay. What an incredible journey we've been on today, diving into this hidden world of bacterial genetics.
It was pretty amazing stuff.
It really is. We've seen how these seemingly simple single-celled organisms are anything but passive. They're not just waiting for slow mutations. They have these 4 ingenious, super effective ways to swap, steal, or borrow genetic superpowers. Direct contact with conjugation, viral couriers and transduction, scavenging DNA scraps via transformation, and even internal shuffling with transposition.
A whole toolkit for change.
Exactly. And underpinning it all, we saw how plasmids are these amazing mobile carriers and how recombination is that final vital step integrating the new info.
It's a beautifully interconnected system, really. When you understand it, this knowledge becomes incredibly valuable.
How so?
Well, these processes together drive that astonishing bacterial adaptation and resilience we see, especially in medicine, with the relentless spread of antibiotic resistance. Understanding these mechanisms helps us grasp the sheer scale of the challenge we face, but also you have to appreciate the ingenuity, the speed and adaptability of life at this tiny scale.
They're definitely not simple bystanders in their own evolution.
Far from it. They are active, sophisticated players, constantly rewriting their own genetic futures.
Okay, as we wrap up this deep dive, here's something to chew on. If bacteria, these ancient organisms, billions of years old, have evolved such complex, rapid ways to share and adapt their genetic code, literally trading survival tools, what does that tell us about the fundamental drive for survival and evolution in all life? And maybe looking beyond biology. Could understanding these tiny genetic deals inform our own strategies, not just for fighting superbugs in medicine, but maybe in engineering, AI, biodesign? Could it inspire new ways to share information, build robust systems, or even speed up learning in our own complex networks?
That definitely raises a broader, perhaps more philosophical question for you, the listener, to ponder. You know, in our world today, with this explosion of information, constant change, complexity, where we're always trying to innovate, adapt, share knowledge faster.
Right.
Could learning from how bacteria so efficiently manage gene transfer offer us some unexpected insights? Ideas for how we might better adapt, share knowledge, build resilience, not just biologically, but maybe culturally, technologically, or socially. The secrets to rapid adaptation might just be thriving under the microscope.